Good morning once again, and this is the last time, the last lecture in this semester. But to my best beliefs, this is going to be a good help for you for preparing for the exam. As you can see, we are going to deal with the recognition, slide recognition for the histology exam. And everything is going to be repetition, just repetition. What I want to tell you that when I was young, ages ago, we had to describe the slide before telling what slide we are looking at. So the classical slide description was using, of course, looking at the slide and telling with your own words what you see. For that one, quite nice to use the microscope, because just with naked eyes you cannot really guess what the slide should be from. Then you start thinking, and you several things you think of, and gradually you exclude most of the slides. Finally, you are happy because there is only one chance remained, and you say, "Eureka! This is the slide, and that is the title of the slide." So based on the description, you think, and finally you get to the revelation of the slide. The only problem is what finally turns out to be wrong. Your definition, your examiner says, "No, this is not the slide," and you can start crying. Now let's see how things are to be said on the exam. Was a sequence of sentences. The first, what you should tell, is the staining. Recapitulation, so everything you are familiar with, but just quickly going through. Most frequently, the hematoxinosin stain is used for the slides. You must know that the basophilia is the same as acidic pH, because anything acidic loves to be bound to basic blue, and the blue hematoxin is the one which is basic, which is alkaline. So everything which is acidic in the slide should appear blue, including nucleus, rough endoplasmic reticulum, and you know so. The other component is the eosin, which is pink, and eosin is acid. It's an acid, so loves to bind to alkaline things. So the eosinophilia is the same as alkaline pH. Most of proteins have got this alkaline pH, so therefore anything rich in membranes, rich in membranous structures, later we are going to get there again. So the membrane-rich structures rich in mitochondria, for example, should appear eosinophilic, should appear pink. The other quite rarely used, some of the slides were stained with passage, so the hematoxin is the background staining the nuclei very nicely, and the pass, as you remember, selectively stains the carbohydrates. So the question you should ask yourself whether the purple color is present or not, and the other thing is that where purple appears, carbohydrate could be present or not. Of course, talking about the goblet cells, the content of the goblet cell is specifically pass stained, and the other is the basement membrane, at least the carbohydrate component of the basement membrane. Here you can see the smooth muscles cells individually surrounded by quite strong basement membrane. Then also quite rarely used is the Azan staining. The connective tissue fibers are the ones which appear blue, and only these one appears blue. And don't forget, not connective tissue, not all the components, only the fibers, and everything else is red. The most important, even if the aniline blue is faded out, the nuclei appear red, so that is a way how to recognize the staining. The other, this one is the ground tooth stained with fuchsin. The fuchsin, as you know, diffuses into spaces, fills up the empty spaces, and that's what is stained by it. The other method is the injection of Indian ink. Few slides, I think 2 slides in this semester, were stained with that. So the vessels or any tube-like structure is injected with the ink, so those structures appear black. And finally, the silver staining. only one slide is showed to you. The silver staining is a very tricky staining because silver nitrate is the one which diffuses into the slide, and after a certain exposure to the silver nitrate is colorless. The slide is washed thoroughly, so everywhere where it's possible the silver nitrate is washed out, and where it remains it stucks into spaces, into capillary spaces with a very small diameter where the silver cannot be washed out. So the slide is developed, physical developer is applied, and metallic silver precipitates in these capillary spaces. So these are the bile capillaries in the liver. The second statement of yours, so after stating this staining, the second is how the organ is assembled. So what's the structure, general structure of the organ? Two chances are. One is when different tissues make up an organ, and that's what you call layered organ, and the other is the parenchymal, when the cells specific for a certain organ is kept together by a connective tissue framework. So the reason why you should state that quite early, because these need different ways of description. If layered, then of course, layer by layer, starting usually from the inner surface, the epithelial lining of an organ, you describe the layers. And when parenchymal organ is to be described, then you should divide the organ, according to what we said before, into parenchyme and the stroma, which keeps in place the cells, and doesn't matter which sequence you start the description. But that is to be followed. Either you start with the stroma. Stroma is not unique for an organ. Connective tissue framework, capsule sending septa into making lobulated the organ, and a micro framework which individually surrounds the cells. And the parenchyme comes, which is specific for the given organ. Either secretory cells, lymphocytes, hepatocytes could belong to the parenchyme. So nowhere else but in that organ or that group of organs this cell type occurs. The layered organs, most of the slides in this semester belong to that category. The layer organs the description should be started from the epithelial lining, as we said before. You must be fed up with these lists, but just let me repeat them. So the tunica mucosa in the innermost layer, starting with epithelium mucosae, sitting on top of loose connective tissue, lamina propria mucosae, and most of the organs have got a third layer of tunica mucosa, a longitudinal smooth muscle layer. This one is the lamina muscularis mucosae. Deep to it comes again a loose connective tissue with all the necessary elements of loose connective tissue. This is called tunica submucosa, since beneath the mucosa it lies. And sometimes, in some slides, you can recognize nervous tissue elements, nerve fibers, pericardia, and altogether they make a plexus called the submucous plexus of Meissner. Deep to it, tunica muscularis comes, talking about GI tract, always an inner circular and an outer longitudinal layer makes up the tunica muscularis. Between them, this one is more frequently visible in our slides. Again, a plexus is to be diagnosed, and that one is the myenteric plexus of Auerbach. After that comes tunica subserosa and tunica serosa if the organ is covered by peritoneum. Tunica subserosa is like the submucosa, loose connective tissue, and the tunica serosa, quite resembling to tunica mucosa but in a reversed manner, consists of lamina propria coming from the lumen towards the surface, lamina propria, and on it mesothelium serosae should be the last layer. It's very good to compare these things. So it's a reversed tunica mucosa is the tunica serosa, with only one exception. It has no lamina muscularis serosae of course, what muscle cells would do, why to move the serous membrane on top of the organ, on the surface of the organ, so that is missing. And deep to tunica mucosa, tunica submucosa comes, and corresponding to that, deep to tunica serosa, tunica subserosa is there. If the organ is void of serous membrane coverage, then the outermost layer is the tunica adventitia, replacing the tunica subserosa and tunica serosa. Adventitia is again a loose connective tissue, as you know. If from inside you must start the description, then starting with the epithelial mucosae, lamina epithelialis mucosae it's also called. The question what type this epithelium does belong to. In case of this semester, columnar or squamous epithelium are the chances, the two chances. The columnar could be simple or stratified, and if it's simple, it could be just simple or could be simple but pseudo-stratified, as you know. The question now you should ask what the surface specializations on top of the cell are either microvilli, microvilli could be, or gynoecilia, and that determines the group of organs where the slide could be from. If microvillated is the columnar epithelium, then equals the must coming from the GI tract. If gynoecilia, then the respiratory or the genitalia, male with simple with gynoecilia, then the female genitalia. Pseudostratified with gynoecilia, the airway passage, and pseudostratified with stereocilia, then the male genitalia should be the source of the slide. You see that both these two systems have got gynoecilia with a specific function, but the difference is that in one case simple columnar, in the other case pseudostratified columnar bears the gynoecilia on top. If stratified columnar is the epithelium, then you can conclude to the urinary system, the urethra. If squamous epithelium, again two chances are, non-keratinizing or keratinizing epithelium. The fact is that keratinizing epithelium only covers the skin, so never should be part of the mucous membrane. But we should mention this because we had slides in this semester covered by this kind of epithelium. So if non-keratinizing is the certified squamous epithelium, then could be the beginning and the end of the GI tract. The beginning, you remember, till the cardia from the oral cavity, and the end, the hemorrhoidal zone of the rectum is covered by this epithelium. The other chance is the vagina has got the same epithelium, and the larynx, where the big mechanical forces act, the vocal fold is covered by this epithelium, and the lowermost part of the urethra, the end of the urethra. Now, while we mentioned the skin, the keratinizing epithelium in this semester, we had slides. One is the lip, the cutaneous zone of the lip. The other is the anal canal, the cutaneous zone of anal canal, and the glans of the penis was covered by this epithelium. But don't forget, then you should not talk about tunica mucosa if that is the epithelium on top of on the surface. Still talking about the surface epithelium, how to make up your mind what type of epithelium you see. Because there are only few slides in which the exact borders of the cells could be seen, therefore always very good to look at the shape of the nuclei. This one determines the epithelium in which the nucleus is. Either oval or round the nucleus could be. Quite easy to understand that there are two types of epithelium having oval nuclei, but the orientation of the nucleus is the one which tells the difference. If the long axis of the nucleus is parallel with the surface, either the free surface or the bottom of the epithelium, that the squamous epithelium you see. If the long axis of it is perpendicular to any of the surfaces, free surface or the bottom of the epithelium, then the cell which contains the nucleus is columnar cell. The cuboidal cells have got always a round nucleus, and as you know, the round, the spherical nucleus could never be cut into different shape, just sphere, smaller or bigger diameter of circle the nucleus appears in. The question what you should a little bit aware of is so pseudostratified, stratified, the difference. People used to say that if in case of the pseudostratified, all the cells breach over the complete thickness of the epithelium, so each of them sits on basement lamina and reaches the surface. That's true and beautiful, but the problem is that you can hardly ever see this without electron microscope, of course. What you can see is the shape of the nuclei within the epithelium, or the shape of the nuclei at the different levels of the epithelium. If all of them have got the same shape, then only pseudostratification occurs. So all of them belonging to the columnar epithelium, for example, but at different levels the nuclei are. But if the shapes are different, so you can see round nuclei, oval below, oval but flattened against the bottom of the cell, even below, that makes the stratification. And that's what you can really see by looking at the shapes of nuclei of the cells. The only problem with this round nucleus that everyone knows by cutting an egg perpendicular to the long axis of the egg, all the cuts appear round, so the oval could be cut into round if cross-sectioned. So in this case, a statistical measurement should be done in your mind. The majority of the nuclei, if they appear oval perpendicular to the surface, then the ones which appear round are having the same shape but perpendicularly cut nuclei they contain. So the tangential cut of the columnar epithelium appears as epithelium containing round nuclei, but this is only because of the level of the direction of the cut. Then you should look at the surface specializations of the cells. Mostly columnar cells have got this one. There are big differences between the three different processes on top of the cell. First of all, the length. You see that below 1 micron is the length of the microvillus. The kinocilium is the next to come with 5 to 7 microns, and below 17 microns, 14, 17 microns is the stereocilium, by this being the longest of the surface processes, except the kinocilium, the specific flagellum of the sperm cell, which could exceed 35 microns in length. Why this sequence came is that because there are similarities between the first two, and the third is completely different from them, even if the length of them is not put in sequence. This beautiful picture quite nicely shows the difference between kinocilium and the microvillus. Just the length of it, if you can see, then it tells the difference. Why I put them into the first two places, because both of these processes are just cytoplasmic finger-like processes of the cells with actin filaments having no ability to actively move, contrary to the kinocilium, which can make the active movement by having two things inside. Engine is needed, so the one which executes the movement, and also needs a small Japanese engineer who controls this movement. So the kinocilia should not irregularly bite, but synchronously they should make a continuous movement in the same direction, synchronous with each other. So the engine within it Within the kinocilium is the, you remember, 9 pairs of microtubules and 2 central microtubules. So with the Danain bridges, everything you should revive in your memories. And the one which directs the synchronous movement of them is the basal body at the bottom of the microvillus, which really synchronizes the movement of them. I hope you remember that the organelle in the cell, which is responsible for all the movements of the cell. Amongst them, the most frequently occurring movement in our body is the cell division. And the 2 centrioles are the ones which pull apart the 2 daughter cells anchored with microfilaments with each other. So the centriole is the one which is responsible for control of movement, and basal bodies are really centrioles, multiplied centrioles. So a cell has got as many centrioles as many microvilli it has on the surface. Surface specializations still compared with each other. Microvilli, stereocilia, kinocilia, just showing electron microscopical pictures to you. This one is a beautiful brush border, so all the processes have got almost the same length, and stereocilia are the most irregular, longest processes, just cytoplasmic processes, of course, and here you can see the kinocilia with quite a regular length of them, and each of them is anchored to its own basal body, multiplied centriole. And the real histological slide, this is the thing what you see on the exam before you saw during semester. This is the beautiful row of microvilli. This one appears as brush border, or because the individual microvillus cannot be resolved by the microscope, therefore it looks like striated border having cross striation in it. And this one is the cell with very long stereocilia, this male genital apparatus, therefore. And here you can see the kinocilia, which are anchored into basal bodies, and basal bodies appear as eosinophilic row below the surface of the cell. This one really annoys me. This is one I don't know. Cannot understand why it has been started a couple of years ago, when basal, anything basal means the same to students, but these are extremely different. Basal body, basal striation, basal lamina, these are very much different things, not even resembling to each other. These are not the same. Basal body, as we just finished, basal body is at the base of the kinocilium. So this one is near to the surface, the apical surface of the cell, just at the base of the kinocilium. The basal striation is at the base of the cell itself, so on the opposite side of the cell, basal striation is located, and the basal lamina is at the bottom of the cell, but outside the cell, so cell sits on top of the basal lamina, so I have no idea how these things could be mixed up with each other. Please don't do that. The basal striation, first you came across with in first semester when the salivary glands were mentioned, or glandular tissue was mentioned, and you remember that striated duct was the name of the intermediate duct of the compound salivary glands, and the name came from this basal striation, which with high magnification and good resolution can be seen at the base of each of these columnar cells of the striated duct. And the other place in this semester you came across with when the kidney was to be studied, and both proximal and distal convoluted tubules had got these basal striations at the bottom of the cell, so facing intercellular space, loose connective tissue space below the cells. In both cases, the basal striation serves the same function, and the function is transport, ionic transport, but it is to be emphasized that this is active ionic transport, so against gradient of the ions, so needing energy, and this energy is served by the rows of mitochondria in between the basal enfoldings of the cell membrane. Everyone can figure out why this structure should appear eosinophilic at the bottom of the cell, because everything is rich in membranes. As we said at the beginning, I told you that we should come back to this point. Then, how to differentiate between the organs of the GI tract. I hope everyone recognizes that this one is the slide, the cross-section of esophagus, and this is extremely frequently mixed up nowadays with the other slide, which has the same epithelium, but almost this is the only similarity between them, and this other slide is the vagina. And again, by not even thorough but superficial thinking, I don't understand how these two slides could be mixed up. Look at the differences. The epithelium is the same, the stratified squamous non-calcareous epithelium in both organs, and in the esophagus quite pale are the cells of epithelium. In the vagina even more pale they are, so there is not an exact difference between the two. Then look at the connective tissue papillae. In both cases, as you can expect, because the epithelium is quite thick, so needing the supply with nutrients through diffusion, therefore connective tissue papillae are present, and present in the vagina as well. Lamina muscularis mucosae, now this is the first big difference. Everyone can recognize cross-section of quite developed thick layer of smooth muscles deep to the deep to the lamina propria mucosae. So this one is the lamina muscularis mucosae in the esophagus, and this is completely missing from the vagina. The other thing is the glands. That's what the main arguments of students used to be when I asked how could they mix up the two slides. They said that because I did not see glands in this slide, therefore this should be the vagina. Now, the glands may be present in the esophagus, but you remember that at only two places, two levels, close to the beginning and close to the end of the esophagus. But most of the slides do contain no glands at all. In case of vagina, never glands are present, as you remember. And the other big difference is the tunica muscularis. Can you see this very thick, extremely thick layer of tunica muscularis with inner circular, outer longitudinal but very strong musculature, and that's what is missing from the vagina, because in the vagina there is a meshwork, a felt-like meshwork of connective tissue fibers and smooth muscle cells with each other. So if you compare them with each other, you can see the main differences. Another thing is the solitary lymphatic follicles, but solitary lymphatic follicles could be present in any of the two organs. So these are the emphasized most important differences between the two. Still, how to differentiate between organs? There are again two slides which people used to be as being hard to differentiate with each other. Why do they mix? Because the glands are found in lamina propria mucosae in both cases. But why not to mix? So the difference is epithelium is not the same in case of the stomach and the small intestine. In the stomach, as you remember, simple columnar secretory epithelium, but in the small intestine, the simple columnar with brush border should be fine. The other thing what at first glance could be mixed up with each other, the gastric pits and the spaces between the villi, the intestinal villi. But if you consider oblique cut of both of them, in case of the gastric pit, if the cut is oblique, then you can see an empty space within the space, if it is centrally cut like here. But if you make the oblique cut through the villi, then these things appear round structure, oval structure, but always with a lamina propria connective tissue core within them. The other difference, the cell types of glands and the cell types of crypts. Here the gland contains, you remember, the 4 different cell types, and in the small intestine again 4 different, but different from those in the gastric pit found in the crypts. This one is a smaller difference because there are only 2 stripes of the stomach when the slide taken from, and the slide contains 3 layers of the tunica muscularis. In case of the small intestine, only 2 layers should be present. Still talking about the same topic, the epithelium, so how to differentiate between parts of the GI tract, really, looking at the epithelium, because if it is simple columnar, then you can make up your mind it must be from the intestine. If intestinal villi are present, then from the small intestine the slide is. Don't forget again the cross-section of the villus, which contains nothing around and contains loose connective tissue of lamina propria inside. The next question you ask when you ended up with this diagnostic point, what does tunica submucosa contain? If mucous glands are within it, then you can tell that the slide is from duodenum for sure. If there is nothing specific, nothing extraordinary, then the slide should be from jejunum. And in case of ileum, aggregated lymphatic follicles are filling up at the different regions, filling up the tunica submucosa, then this one should be taken from ileum. If there are no intestinal villi, then of course the slide should be from the large intestine. Question whether crypts of Lieber can make difference or not. They are both found in small and the large intestine, so making no difference, but the cellular components of the glands are the ones which make the differentiation. In the case of the small intestine, you remember Paneth cells to be present, but in the case of the large intestine, there are no Paneth cells, no need for digestion, no need for enzymes to be present. Therefore, mostly goblet cells, enteroendocrine cells, and some germ cells should be found in the crypts of Lieberkühn in the large intestine. These are the 3 slides, again quite frequently mixed with each other, but you should. But the reason is people mixing because the epithelium is simple columnar epithelium, but in this case, on top of the cells, there are kinocilia because the row of basal bodies is visible. In case of these epithelial cells, the cells, all of them secrete. And this secretory function is indicated by the pale eosinophilic cytoplasm of the cells. And finally, on this one, in this slide, you can see a very thin layer of cuticle, the brush border on top of the cells. When you look at the lumen, this again makes difference. In case of the first slide, there is only one lumen in the slide. In case of that one, similar, but 2, 3 more than 1 lumens, and again only one is seen in this case. And the recesses you should look at now. Closed recesses, it means that spaces where you cannot really reach the main lumen, main passage of the organ in the level of the cut at least. You cannot, in other cuts of course they communicate with each other. So in case of this slide, there are no closed, no hidden recesses, always free way is found to the main lumen of the organ. There are recesses, closed looking recesses in this case and that case also. If you look at these differences, you can make up your mind that must be from uterine tube, this must be from seminal vesicle, and the last is from the gallbladder. So no more mixage will be happen, I hope so, by considering these differences. Then parenchymal organs, you should look at the difference between them, and of course the difference lies in the parenchyme specific to the given organ. If the sporenchyme is made up of secretory epithelia, then you can state that the slide is from a gland. But the two different types you had to distinguish in this semester, the serous and the mucous. We should get back to this point. If lymphocytes, then what a wonder, lymphatic organ you should look at, and you should differentiate between them. If there is aggregated lymphatic follicles in mucous membrane or covered by epithelium, this is the same, then the tonsil, the definition of the tonsil arrived. If the same aggregation of lymphatic follicles, but surrounded by connective tissue capsule, then appears to, that is characteristic for the lymph node, so you can say that. If there are lymphatic follicles, but in general you can differentiate between two pubs, white and red, so two different substances that characterizes the spleen. And if there are no lymphatic follicles, but the organ is lobulated, so the units are lobules, then the thymus you are looking at. In other cases, quite trivialities, so quite easy to understand that seeing hepatocytes in a lobular structure, that equals to the liver. If you see follicles at different stages of maturation, not lymphatic follicles, don't take me wrong, just follicles, then equals to the ovary. And if you see male germ cells at different stages of maturation, then of course the testis, the slide is from. Then, again, frequently mixed, you can recognize two glands, but the staining is different. So one thing is the cytoplasm, in this case, is really blue, so basophilic, same with hematoxin, but in this case, this is purple. And this is the color, you remember, which was characteristic for the past staining. So by looking at this, you can see that this one is differently stained than that one. So further investigation, if you make, then look at the shape of the nuclei. Here in this case the nuclei are more or less round, and here the nuclei are flattened against the basal surface. And if you look at the position of the nuclei are more or less centrally located here, and here at the surface they lie. And this is the reason why this one is a serous and this one is a mucous gland, but with past-age staining. So don't never forget that one sign is not a sign. So if out of three only one fits, then it is not enough to say that it's serous because the round nuclei I'm looking at, but you should take into consideration more than one facts. The lumen is also a different thing in these two cases. Here you can hardly see but you remember this one is a repetition and quite visible or relatively bigger lumen, the mucosa acini have got. Also you remember the reason for that. Mucous product is rich in carbohydrates, and the best example is to think of honey, for example, which is a thick solution, very thick solution. So carbohydrate is the one which needs wider lumen to pass through, and the serous solution is rich in proteins, enzymes, so quite thin is the fluid, so smaller lumen is enough to pass through. Another thing which is frequently not known, it is a beautiful illustration taken from the book of Professor Kristich, showing the renal glomerulus. One can recognize the podocytes, the specific processes, the glomerular slit membrane and everything here. And the big question what you should ask, because finally the kidney should be considered as a big filter, of course making ionic exchange after the primary filtration. The primary function is the filtration of the blood, and the urine, primary urine, is produced into the cavity of the Bowman's capsule. So the most important question is to ask what the filter, the ultrafiltrating layer, what the filter paper in the kidney is. And the answer is the unusually thick glomerular basal lamina should be the filtration material, because the fenestration of the capillary endothelium does not prevent molecules to pass through, and the same applies to the slit membrane here, where the spaces are still fairly big enough to pass molecules, medium-sized molecules through. So the real filter paper is this glomerular basal lamina. As you know, by making coffees for yourself and filtering through a paper time after time, the filter should be thrown away. So here in the kidney also, the filter paper should be replaced, because materials, molecules stuck into it, and finally nothing could penetrate through. So the question, what the basement lamina is produced by, and the answer is that the podocyte, the function of the podocyte is continuously to produce the basement lamina. And the other thing is, there is something which should remove the used filter paper from the place, and the function of this is the function of the specific macrophages, the intraglomerular mesangial cells. Now, after this last sequence, nothing is left but to wish you very good luck for the exam, as well as good health to survive this pandemic horror or something. Be strong. Goodbye.